Welcome, everyone. We're here today for a webinar for the Statistics Former Student Network of Texas A&M University. We're very fortunate to have Dr. Veera Baladandayuthapani speaking today. Veera received his PhD in statistics from Texas A&M University under the supervision of Raymond Carroll, Bonnie Malik. His research interests explore the potential of Bayesian probabilistic models and machine learning methods to assist in medical and health sciences. These methods are motivated by large and complex data sets as high throughput genomics, epigenomics, transcriptomics, and proteomics, as well as high resolution neuro and cancer imaging. He's currently a professor of statistics or biostatistics at the University of Michigan, where he also serves as the director of the Cancer Data Science Shared Resource. He's received numerous prestigious awards that include being selected as the Murdo Leftopoulou Distinguished Lectureship from Harvard School of Public Health and the H.O. Hartley Award from the Department of Statistics at Texas A&M, as well as the Young Investigator Award from the International Indian Statistical Association. He's a fellow of the American Statistical Association and an elected member of the International Statistical Institute. He serves or has served on the editorial board for major biostatistical journals such as the Journal of American Statistical Association, Annals of Applied Statistics, Biometrics, and Sankhya. So welcome everyone today. Veera, I think we're ready for you to get started. He's going to give us a presentation on drugs, data, and me. I'll let you share with us more about your talk.
Yeah. Thanks so much, Marcy. Thanks so much. That was a very generous introduction. And I was just scrolling through the participant list. It brings back so many memories of my time at Texas A&M. So really delighted to speak here, and thank you for the invitation. Let's see. I'll go to, Marcy talked to a little bit about it, but I'll just give my point of view and some of my Aggie experience. I worked with two outstanding mentors in Ray Carroll and Bonnie Malik, and my dissertation focused on doing Bayesian complex model with cancer applications. I've had the privilege, being in Texas A&M, having a very outstanding and generous professors. And I see some on the list here, so it makes me a little sheepish here, and also peers, and those days, Blocker was literally my second home. So very, very fond memories, especially of the vending machine, if it still exists in the basement. And of course, I met my wife there as well. So it has a very special place. No. Oops. Yeah. So so just to sum it up, look, now I was told long ago that a very good way to judge how well you're doing is to measure the quality of people with whom you interact, and by that criteria, I could not have asked for more in my time. So, and at College Station in Texas A&M. So I just wanted to. This was my first ever seminar flyer, and Elaine might have created this a long time ago, 2004 it was, and this was my practice job talk when I gave a seminar in the department, and that was my first ever seminar, and I was giving it to all my major professors, mentors, and my thoughts at that time is pretty much characterized by this PhD comics, how nervous I was, when I was giving that talk, and it was right after my thesis defense. So I'll just give a couple of seconds for you to read it over. I pretty much cobbled it up as I was moving along. Well, I hope there's a chuckle somewhere behind all those mute mics, but on that note, had some wonderful memories and times, and I'm glad to circle back, after so many years. So getting over to the scientific portion. So I wanted to set the stage of a more general kind of talk, balancing, aspects of what I've been working on both in my graduate school as well as I used to be a colleague with Masi at MD Anderson, and working on biomedical problems, and zooming out, giving it a broader perspective, the promise of precision medicine and how to appropriately cast it, and what's the role of statisticians and more generally quantitative scientists in this area. So I was doing a little bit of a deeper dive into it. 1 second, sorry, I'm trying to hide this panel. Okay, there we go. Into what goes into this concept of precision personalized or stratified medicine. So it sits under a broader framework of what's called the P4 medicine, where the 4P stands for different aspects of medicine. That includes predictive medicine, preventive medicine, what we know, personalized medicine, as well as participatory. And this sort of stems about by of course technological advancement, both at the clinical level, but also through molecular biology and the data streams that come across. And predictive, just parsing them out, it tries to contextualize the types of scientific questions we aim to answer and what sort of quantitative needs, challenges that motivates. So predictive would be identification of individualized risks based on the particular genetic profile. Preventive is on a more a backward step in terms of trying to avoid or reduce, and then personalized goes into more the clinical interventions, the aspects of treatments and drugs and therapies, and that's where my talk would be sort of situated on. And participatory being, going the other side, where the patients are fully engaged in their own, and that's also becoming an increasing concept in this P4 medicine framework. So that of course comes with a lot of opportunities, and this phase actually accurately characterizes my feelings when I was a junior faculty at MD Anderson, and actually when I walked into, when actually Keith Baggerly and Kevin Coombs, my ex-colleagues, described me this whole TCGA project and the types of data they were collecting and where the field is moving, in collecting data at different omic levels, genomics, transcriptomics, phenomics, proteomics, and what sort of opportunities are there. And that essentially, giving them full credit, kickstarted my career, at least into thinking about those problems and making some contributions. Of course, there's the opportunity, but there's of course the challenge. And the challenge of putting these different sets of data together, can be parsed out. What are the specifics of the types of questions or knowledge that we can gain? It includes both extracting biological data, biological knowledge, as well as and contextualizing them what might be clinically implementable using that knowledge. So it goes from more discovery as well, but also keeping an eye on what might be the eventual the clinical translation of what we can gain. So fixating on that axis. And this might not be an understatement that now that with technological advances and the types of data, the different scales of data that's being collected, that data integration may be the key quantitative challenge in driving modern scientific endeavors, and that includes both biomedical but as well as other fields. So I've worked in cancer throughout my career, both at the graduate school at Texas A&M as well as when I was at MD Anderson. So I'll use cancer as the context here, the scientific and the clinical context here. And cancer has, as many of you know, has many bad things going on for it, but it also happens that it is actually one of the most well-characterized path physiological and path biological. What that means is, there's so much funding gone towards cancer and understanding the basis of cancer, and there have been many aspects of the disease that have come through over the last decade or couple of decades or so. There's still a lot to learn, but what that has done is, it has germinated multiple types of high throughput data that are available on matched model systems. What I mean by model systems is not only patients, but also which is the key, the patient-derived samples, but also what are called preclinical model systems, such as cell lines, patient-derived xenografts, which are sort of used in a preclinical laboratory type setting for drug discovery and such. And this is something that's growing day by day. This started off with national consortium level effects, because these data are not cheap to collect and requires a lot of infrastructure and funding. And so the TCGA is sort of one of the flagship consortiums in the US. There's the ICGC, which is more international, which has all been collected into the National Genomic Data Commons. And I would say 10, 15 years ago, this was done at a consortium level. But what's happening now is also at institutional levels. Now folks are starting initiatives, and I can talk from a Michigan perspective that we have something called a Precision Health Initiative at University of Michigan that's collecting a lot of such data, multi-omic, multimodal data on patients, and building what are typically called biobanks, collecting different types of not only genetic information but all of the clinical history and other aspects as well. So that brings about a plethora of really nice questions and quantitative questions that motivates. So I want to contextualize before I get to the meat of the talk. I want to contextualize, what precision medicine in cancer, which turns out, precision oncology, where it was and where it's moving to contextualize some of the methods that we have been working on, not only myself, but with collaborators and a lot of folks in this area. So where it was like around 5-10 years ago, what can be coined as precision oncology 1.0 is based on a small number of molecular abnormalities. So it is using the underlying molecular or the genomic information from the cancer samples, but it was based on a few. So I would say maybe a few molecular abnormalities, and always constrained by the tissue of origin. What that means is if it's breast cancer, it is breast cancer, it's kidney cancer, it's kidney cancer. So where the organ site was. So sort of where the current is, it's always the field is evolving, but where the current is going beyond the small number, which could be a dozen or a few, it's now gone to these dozens or even hundreds of possible what are called mutational hotspots, genes you can think about. And these are cancers that are cancer-associated genes that have been discovered over the years. And there have been this has actually drawn a plethora of companies doing these what are called panels that are now used and both in an academic setting as well as in different industries as well. So now what's it done is it's gone beyond being the tissue of origin. What that means is going beyond the organ side, but could be instead of treating treat the cancer, instead they treat the genes. So that could be the same gene could be across lung cancer, brain cancer, or kidney cancer, just as a conceptualization. So there's a very nice review article I came across that sort of says, okay, what might be the 3.0 and what things we should be thinking about, what aspects go into that. So this paper actually has a nice sort of a life cycle here that not only goes from collecting the samples, going from panomic data, doing an analysis, but piping it back to what might be the treatments. So the aspects that go into it are panomic analysis, and statistical reverse engineering, essentially, finding more from a discovery standpoint, what can you glean from integrating such data, and what might be driving these tumor cancer processes, which are called oncological processes. And so my talk actually, what I'll be talking today is sort of going through this cycle, and I'll just give you flavors of what sort of problems come about that, and that helps what sort of methods what we can develop, either standard methods, reorienting standard methods or developing new methods to answer specific questions. So from a network perspective, I'll go a little bit into network-based models for these panomic data integration problems, and one particular sort of pet project of mine, what I call personalized networks, and I'll make it more clear as we move along now. So I've sort of bucketed them into three different parts. In the second part, I'll talk about some earlier work, earlier and ongoing work on targeted discovery. It's on targeted discovery of finding genomic markers, what we call the IBAG model, the integrated Bayesian analysis of genomics model, and sort of some generalizations and extensions and of that, especially with imaging data. And finally, now I'll talk a little bit of our current work, what our focus is, and sort of a problems that sort of has consumed me over the last, I want to say, a year or so, is how do we go from discovery to actual translation, and how do we pipe it back to drug targets and drug discovery or drug, and that's a field that's called pharmacogenomics. So it essentially turns out to be a problem where you integrate data across different model systems, and from a statistical perspective, you can think about as different populations that can include patients and also lab-based systems and for drug response calibration. So as you might imagine, each of these is a talk of its own, and I have to pack all this in what, 40 odd minutes from now. So my idea here is to give you a flavor here without going into the too deep technical details, but I'll be happy to chat. I've put in the references as well, and hopefully I'll be able to do justice. So let me start with networks or graphical models, and it's Bani Malik, my major advisor, introduced me to this field when I was towards the back end of my PhD, and it really caught my fancy because I feel networks are something that sort of become a common alphabet sometimes when you speak to clinicians and scientists, because everybody understands networks. You lay out different nodes and edges and connections, and it makes very intuitively a very nice, sort of a common way to speak and show them results and interpret them. So the basic premise, at least in a genomic context, is that most of these, if you think about a network, which is we have different nodes and different edges, and the nodes could be genes or proteins or any of its products, and you model regulatory or association relationships. So association means it's correlation type, or regulatory could be more directed, and that has a bit more different interpretation in terms of causal mechanisms. Although causality is a own beast in itself, and I won't get there, but more directed versus undirected edges. So networks, I mean, it's a lot of brilliant people have worked on it over the years. But when it comes to building networks across different types of omic data, that opens up a lot of different types of network problems. That not only includes undirected, directed, as I said, but also something called chain graphs, where you want to model relationships between these omic levels. And in terms of inference, how does the graph change across populations, heterogeneous graphs? So how does change with populations, or testing for changes, which is differential graphs? And I'll talk about patient-specific networks in a minute, and also building prognostic models with networks. So essentially, just to quickly summarize, networks are useful for these higher order organizations. Instead of looking at one or two genes at a time, you're looking at a more systems level, and that helps identify certain components of the network that might be driving biological mechanisms. In the context of cancer, that might be carcinogenesis. So that was just setting a brief primer on networks, and when you go into the data integration realm, there could be different types, as I said, of networks one can build. So it could be, vertical data integration. What I mean by that is, you have the same type of cancer, but different types of omic data. So copy number is typically what's called DNA level data. Gene expression is what is called transcriptomic data. and this is methylation data, now another type of omic data. And what you endeavor is building some sort of a network that connects, that shows different types of regulatory patterns. And that's something Minjin Ha, a former postdoc of mine and now an assistant professor, developing these type of networks for proteins and genes across different cancers. Or it could be horizontal data integration, where you not only have data now for a given cancer across these modalities, but you could have it for multiple cancers. So you could have a common network, or you could have a different network. And I know one of the faculty, Yang Mi, Texan, has worked a lot in this area as well. So getting into the connection of network models and precision medicine. Now, how do we actually personalize graphical models? So, so one thinks of networks, it's a population level summary. So usually when we think about it from a statistical perspective, it's usually a covariance or a precision matrix. And when you get to patient-specific networks, it's inferentially what we are trying to do is it's sort of a ill-posed problem, in the sense that you're trying to decipher a network at an individual level for n equal to 1 using p high-dimensional features. So that you can think about like hundreds of genes, or on that order or even more. So in genomics, typically we don't have within patient replication. What that means is, like, genomics data are hard to get, so you don't actually repeat the experiment multiple times. So you only observe at one snapshot. So how would you create a network that can give you networks at the individual level? So this is a problem. We've been thinking, a lot of other folks have also been thinking what might be. So there are two possible routes as I see it. Now, one could be a supervised manner. You do need some other additional source of information. So, for example, a supervised approach would be graphical regression, where you regress a network on some other information, which could be the patient-specific covariates. So the patient-specific covariates, any additional specific information which tells you the heterogeneity of the patients. And then in that case, then one can and I'll come to it in a minute, like how edges and hence structure changes with the covariates. Or one could do graphical deconvolution, where you say, okay, you get a population level network, and then use any additional source of information to get patient-specific networks using some context-specific information, but doing it from the top-down end, while getting the network. And show you a couple of examples of those methods as well. Right? So to just fortify the concept a little bit, now I used to work a lot with hematologic malignancies, which is basically blood cancers, when I was back in MD Anderson, and still do at Michigan. One particular type of blood cancer is called multiple myeloma. It's a myeloma. Multiple myeloma is sort of a blood cancer that develops in the bone marrow, and it typically goes through multiple stages. What's called MGUS, which is the earliest form, and then goes through a series of what is called smoldering, symptomatic, then myeloma, where so these two are asymptomatic, it's not detected, and then it goes to symptomatic. That's when you see the symptoms and treated. It's very treatable when detected at the early stage. But when it gets to the relapse refractory setting, it becomes much more harder, although the treatments have evolved a lot over the years. So typically how it is staging means like how it is broken down into categories of myeloma, which determines what sort of what treatment protocols to give. It's usually done by two biomarkers. One is called serum albumin, and one is serum beta 2 microglobulin. So essentially, it's a blood-based, from the tumor, and then you get these two proteins, and if the level of the proteins is in the green zone, it's called a stage 1. If it's the yellow zone, it's stage 2, and if it's in the pink zone, it's stage 3. My apologies, the lawnmower guy just came for the next door neighbor, so you hear a lot of buzzing noise. Hopefully it'll be done soon. So based on the two biomarker, say it is broken down into different stages. So if you think about it, from a patient perspective, you can break down the patient, a group of patients into three separate buckets based on what the staging is. And then you can see, okay, what determines, what sort of molecular behavior changes across these patients. So, from a network perspective, that would be, basically saying, okay, these networks change across these different stages. But if you look at like two individuals who are right on that edge of the boundary where their values are very close together, but they're actually at different stages, you would assume that these two individuals would have some commonality in the networks. So that led us to think that can we blur these boundaries and think about this whole process as some sort of continuous disease evolution and build these networks that can evolve, as a function of this. And that's basically at the essence what the concept of what a personalized network might mean, that individuals at different levels of the disease could have vastly different networks, and how do we decipher that using data? So this work actually was led by Yang Mi, and he's been a leader in this work through and through, who's actually a current Aggie now, who developed during his dissertation this method called Bayesian graphical regression, which essentially does the following. It essentially posits graphs or covariance values. It takes the network and regresses it, the latent regression on the covariates. So what it means is, from a statistical perspective, is how edges change with covariates, how particular network edges, like you have gene 1 and gene 2, how does the relationship between them change with covariates? And that leads to, when you do it at different values of X, you essentially get patient-specific networks. So you could do either for directed networks, associate correlation type graphs, or you could do for, directed or undirected as well. So our initial work was focused on directed, but we've extended to undirected as well. And the other is, you don't expect the associations in biology not always to be linear. You could have non-linear associations as well, and how to bring about linear, and as well as to graph structure prediction. So, and I wouldn't go too deep into the model construction. So Yang's paper, at least the first paper in this regard, has come out in just a few years ago, that goes into the mathematical construction of this and the Bayesian formulation of this. But I just wanted to give you what goes in and what goes out. So essentially, if you start with a particular, in multiple myeloma, a particular pathway, you have a set of genes, and essentially you have an N cross P matrix of genes. And then you get information on these staging biomarkers, which are values, so those covariates, and then you can get networks, which can tell you, what is the relationship between two genes, right? And how is that governed by different. So Yang developed this. So going a little deeper into the types of relationships. So there could be relationship between two genes that is not impacted by the level of the covariates, which in this case was, the two staging biomarkers. Or you could have, they could have a sort of a constant, change, and they could have a positive constant change, or the regulation might be, a positive. So in this case, if this gene increases, this gene, so it's a negative association, negative or positive, depending on the deregulation or upregulation. And then you could also have where it could be varying linearly with the covariates, or it could be non-linearly varying with the covariates. So there might be a biomarker which, relationship between two genes at particular, stages, it might have a different relationship, but at different biomarker, it could be different. And how is that whole thing changing? So in this context, we looked at, 5 critical signaling pathways, in this case, from multiple myeloma, to look at these, and we had some nice biological results and interpretation now how to set this. And Yang actually built a very nice shiny app as well, where one can visualize these results, where the link is given here. I'm not going to run it, but basically you could change the different prognostic factors, the 2 staging biomarkers, and you can see how the networks change across, and nicely says, what different relationships can be gleaned from that. Right. So So from a graphical deconvolution perspective, so this is what I was meaning, the unsupervised way, where you can build a graph. So just to make it a little bit more clear, so this is a method called Precise that was developed by Minjin Ha, who's now who's an assistant professor at MD Anderson. So she built this method. We worked on this method where we took omic data from multiple cancers. Okay, we looked at around 30 cancers. So what we did was we built what we call a cancer-specific network. So BRCA here is breast cancer, KIRC is kidney cancer, UCAC is uterine cancer, and we incorporated what is already known. That there's a lot known about these genes and proteins what are stored in databases, and then and using a causal net, basically a directed network type modeling, we came up with a cancer specific network, and from the cancer specific network we deconvolved a subject specific network, and to compress that information into sort of what is called a single number summary. Is this so you can think about this whole network as a pathway, and come up with pathway level summaries, whether is that suppressed, activated, or neutral. And once you get this, so this is learned in an unsupervised manner, then they can be used for subtype identification or types, different types of cancer, or you could do, if you have clinical outcomes, for example, survival or prognosis, you could correlate, associate them with outcome prediction, and subsequently also do validation. Again, this the results and code and everything is on this shiny app that Minjin built. So just to go into a deeper, how do we actually do it? So essentially, under the hood, it sits on in a sort of a Bayesian regression. Essentially, it's a Bayesian estimation where you regress each of the nodes on all other nodes, and then using a variable selection prior, we get a sparse network at the cancer level, and then using the predictive distributions, the posterior predictive distributions at each of the nodes, one can bucket the individual nodes into either it's neutral, activated, or suppressed. So you can get the network at that level, and then you basically combine it now into a score, where the score is determined by how many upregulated genes are there, how many downregulated genes are there, which is weighted by the number of genes it controls. And that's the utility of bringing in network information, because once you get network information, a particular gene and how many genes does it control. So if that gene controls a lot of other genes, then it becomes an important gene, and that gets a higher weightage, as opposed to some other gene which is a lone ranger, and that gets down weighted. So essentially, we got these scores, and then we correlated it with. So we get 3 types of scores: suppressed, activated, and neutral, and for each patient at a patient level. So these will be at a patient-specific level. Right. So this idea of doing this came about with my collaboration at MD Anderson with Gordon Mills and Han Liang. At that time they were collecting what is called RPPA data, which is proteomic data on the TCGS samples that included around 32 cancer types, and and some of the cancers, for example, breast cancers, the more prevalent cancers had a high sample size, but there were some with very low sample sizes. And for this work, we focused on key signaling pathways as a proof of principle. So we looked at, these 12 pathways are very important pathways in cancer, and a lot has been studied on them. In terms of what is the mechanism behind these pathways, the mechanism also, and these are very what I say clinically druggable pathways. So anything that we can glean in terms of what's going on in terms of these pathway activities can be highly informative in driving some biological insights. Right. So there's a lot packed into this figure, but I'll just highlight a couple of quick things here. So across these 32 cancers, multiple samples, I think we are around 7,000 samples. What if you focus on this one, this heat map, what is showing you is that scores that I mentioned, the precise scores, that network scores. So we got the precise scores from, so along the columns are the patients, and then along the rows that you see are the 12 pathways, so each of the network-based pathways. And we got the precise scores, and we just ran a simple clustering on it now. And the couple of interesting things that we found, that using our network scores, we found basically 23 optimal clusters, and each of them, so that is this row right here, which is the clusters we found. And we layered that with what is the tumor type of origin, and which is not surprising that there's a high fidelity to the tumor site. I mean, you would expect that different cancers would behave differently, but what you can also glean is what exact pathways contribute to that. How do those change? And while that is a more global result, but at a local level, there are different clusters that have a very different mixture of cancers now, and that have very different types of pathway activity. So a couple of would be C5 right here and C16 right here, which is a mix of multiple different types of cancers. And also we found that breast cancer, which are the highest sample size, that broke into 3 different types of clusters, and based on different types of pathway activities. And these 3 groups had very different prognostic patterns based on these pathway activities. So, and the bottom panel here, I won't go too deep into it. Basically, it's concordance with established mutations, and which is now, as I said, the 2.0, which is based on mutations, and there's a very high concordance with the mutational hotspots in this. So it was a sanity check, and B, what complementary information can we get from proteomic data by this analysis. And one more thing we noted was the clustering. If you cluster the raw data, it's just not as clean as just our PPA data. So it aids 2 ways. It brings out the pathway level signals a bit more clearly, and at a P level, it's also very interpretable, because now you're bucketing the proteins by different biological mechanisms and trying to understand them. And we have a host of other results in the paper as well, in terms of specific network attributes that we looked at. So one more thing we looked at was, I won't go too deep into it, essentially, how does it well in predicting prognosis? So this is what is called a spider plot. So what this is plotting is for each cancer, each of these lines is a cancer, and these are the p-values of associations with survival for different pathways. Now, and for some pathways, there is very high degree of association, for certain cancers, and we compare it to other approaches, existing approaches, for our PPA data, and we found that in general, we find higher prognostic power as compared to other approaches that don't take into network information. That being the punchline here. Right. So before I move to part 2, I'll just do a brief pause here if there are any questions. No. Sorry, I can't see the chat screen. So if there are any questions, Marcy, Elaine, let me know. I'll be happy to take them as I roll along.
There's nothing in the chat right now.
All right. Yeah. And please feel free to stop me anytime. No, it's going in at a high level, but I can at least impute some information as best as I can. So the second part, I just want to talk about some more targeted discovery approaches and talk about this IBAG model. And again, I'll give you the conceptual underpinning and eschew some of the technical details here. Now, so the conceptual framework of IBAG is a little different from networks, but so when we started thinking about putting this data together, when you have different types of omic data, So a nice way of thinking about this, or a more tenable way of thinking about this, is that the genomics tells you something about the mechanism, what's going on, either at the transcription level or at the different DNA level or at the microRNA level. And then what you eventually observe is the clinical outcomes. So the way we thought about this is parsing down into two models. One will be a mechanistic model that learns the mechanism. One would be a clinical model that learns about the clinical responses and basically put the two and two together, right? So that's sort of the construction. So to make it more, mathematically or statistically more precise, so suppose you get, high dimensional data, call it xmRNA, which collects all the genes by samples, and then you have xDNA, which is collecting the genes and same genes, but the DNA level information on those genes. And then you have microRNA, and all of these matrices are sort of row matched in the sense that they come from the same sample. In this case, it will be the patients, but it will come from the same. And think about a clinical outcome, for exposition, just think about one outcome, say scalar, think about survival. But they could be anything. They could be multivariate, or they could be functional, even an image. And that's something that we've worked on as well, right? So what we do is we want to build, from a Bayesian perspective, we want to build a joint distribution on this joint data matrices together. So what we do is we parse them into two, depending on the expression, using the expression. So we have expression given the upstream elements, which we call the mechanistic, and then which has some model parameters, which I'll make it clear in a minute. And then you associate them with clinical outcomes, right? So why do this? Why break it down? So it's actually turns out from the biology of cancer. So what happens is, when cancer evolves, that one can have different levels of DNA level changes, and that DNA level changes can be inherited, through your parents, or there could be some aspects that you're exposed to, and that can be very heterogeneous. But what it happens is these different DNA level mechanisms coalesce into transcriptional mechanisms. And that transcriptional mechanism, and many people have found this, is surprisingly coordinated for different cancers. So what are called transcriptional programs. So these are specific mechanisms, and these are highly conserved across samples. So at the transcriptional level, that's why we use transcriptional level as the eventual level which correlates with the clinical outcome, because we want to find those programs that are conserved and how did the different DNA inform that transcription. And so it's usually basically using the basic principles of biology by going from DNA to mRNA. Of course, there's a protein level here, and then eventually so it's basically mimicking that hierarchy, but at the same time has a nice interpretations of what those states might be, right? And also, of course, that biological. So that's what I mean by a dimension reduction using both biological and statistical principles. So what that means is by breaking this down like this, it's biologically justified, but from a statistical perspective, one can posit any model, but if you do regression, which is at least my favorite model, that way you can put very flexible models on that, especially for the clinical model, as I said, because the clinical outcome could be of different scales that you can accord here. All right. So this work actually started, a few years ago with Jeff Morris and Elizabeth Jennings, and they both are former Aggies. Elizabeth graduated from Texas A&M, working with Ray Carroll, myself, and Jeff Morris. And Jeff, of course, many of you might know. And actually, I was just on a call with him. He told me he's giving a talk in December, so a fantastic speaker. So I'm just giving a plug for his talk. So how do we parameterize the mechanistic model, like I said, it's a regression model, but a nonlinear regression model, where you take a gene and you just regress it on the upstream DNA factors. So those could be the DNA, methylation, and microRNA. So the key idea here is, if you observe a gene expression, you break it down into different components. And essentially, you can view this as a denoising procedure for estimate of expression. And what that helps is, as I mentioned before, there could be a lot going on in the DNA level and at the upstream level, and then one can identify driver genes, what actually drove these expression levels, either drove up or drove down, or different kinds of relationships, and and finding those genes, and then using the components of these different components, you build in another model which is the clinical model formulation, where you bring in these different components and then you associate with the outcome levels, right? So the mechanism is learned at the gene level, but then the clinical model integrates that information across all the genes using another model at the clinical level, right? So so this seems an overtly complex model, at least at one level, in trying to understand the basic, the basic systems level understanding. But there's a very nice interpretation of the parameters, in putting these models together. So when you learn something from the mechanism and put it into the clinical model, these parameters actually have a very special meaning and very interpretable meaning. So as I said again, so I'll dial back a little bit, and so basically what happens in cancer is basically one cell gone wrong, right? Like one cell gets mutated, it talks to the cell nearby, the other cell gets mutated, and the whole process. So how did the cancer cell go rogue? So think about, like, inside a cell is your DNA, and there's some kind of a switchboard, right? That's the cell has an epigenomic switch, it has a genomic switch, or possibly a transcriptional switch. So think about a circuit inside the cell. So what this model is trying to uncover is what switch went on or off inside a cancer cell, or in this case, it'll be a collection of cells because it's a sample level. It's not single cell data, but and if those cells, what are the effects that are modulated by methylation? That will be an epigenomic switch or a genomic switch, and then and those effects, how do those effects manifest as an effect on the clinical outcome? So if you think about regression, so these are regression coefficients. So if this is high, so this switch got turned on and it had a positive effect on prognosis, right? If it is positive, it is negative, then the switch did get turned on and had a negative effect. So, and it doesn't have to be mutually exclusive, because biology is complex. You could have multiple switches turning, for different genes that might confer positive or negative, if you think about why a survival prognostic effects, right? And so basically, this one is the biological interpretation of these models, but also, how do you actually fit these models, and how do you actually borrow strength from these different levels of data? So essentially, in this case, we use what is a class of shrinkage priors called normal gamma priors, where you can have different formulations, to borrow strength across genes. And that has, and some work in this area, we have done and others have done as well, that can allow, I guess the name of the game here is shrinkage, and shrinkage is, you can model better tail behavior as well as, computationally easier to fit. Let me keep a track of time here. So so what goes into IBAG? I guess I've so at a high level, so in the IBAG model fitting, you have two steps when you have a mechanistic model and a clinical model. And as I said, each of them has their own specific interpretation. And we actually have a Shiny app also to fit these models now. And a student of mine is actually working on, just jazzing that up a little bit and making it so not just a Shiny app for visualization, but can we make it fast enough computationally so that folks can use it on their own machines? I'm going to skip through the results a little bit and just whiz through. I see I have just 10 minutes left, and I want to keep some time for questions. So we have done several. That was the one of the first IBAG works, but we have done a lot of work in extending these models for both linear and non-linear settings, and also incorporating radiologic imaging as well. One of my PhD students, when I was back at MD Anderson, she worked out what we call radio IBAG to have not only DNA level data, transcription level data, but also radiologic imaging biomarkers and outcomes. And a student of mine is right now working on incorporating proteomic information. And that's the link to the Shiny app. And I'll be happy to circulate these slides so that you have those links. All right, I'll skip this, the pathway IBAG. And just whiz through in 5 minutes about the translational aspects, because I think this is a very cool area and something that has me chuffed, keeps me up at night. So this is looking beyond, like right now, initial part we only looked at patient samples by themselves, but then going from discovery to translation, how do we actually go towards drug, incorporating drug level data? So the high level goals of this work is, you have patient level data, across these different consortium level, from these public databases. You have cell line data that's been collected and which have been profiled at the multi-omic level. And corresponding to the cell lines, because these are laboratory based systems, and there's been a lot of work, by different groups in collecting drug data, trying to perturb these cell lines and see what drug works. And essentially, the idea is that if a drug shows a promising mechanism in cell lines, they can be translated into patients. There are a few more hoops to jump there, but that's the idea. So this integration is a little of a different variety, that you have omic data, but you have data at different levels from different sets of experiments. So the idea is, can we exploit the conserved biology between cell lines and patients to calibrate what might be a therapeutic response of drugs in patients? And that innately becomes a quantitative question, is, how do we integrate data from the cell lines, and the corresponding drugs on the cell lines to inform, not necessarily in a prescriptive manner, because of course we can't do that with models, but more at a suggestive angle that these drugs might be more promising, And historically, these cell lines have been, the cell lines that are used for testing drugs. It's sort of in a very subjective manner, in the sense that depending on availability of the cell lines or what cell lines, but can we improve the identification of cell lines who are better proxies for patients? Because we have this patient data, can we incorporate this, saying these are the major cell lines that have more closer resemblance to the patients, at least at the genomic level, and they could be prioritized. And then, of course, what is the genomic drivers explaining these cell line similarities? That'll help as well. So statistically, in a broad context, this becomes building joint probability models across patients, cell lines, and drugs, and it's actually a very exciting area, I feel, and I have a current PhD student who just worked on extending the precise pipeline to incorporate cell lines as well, to do drug sensitivity prediction. We got a JCO paper, Journal of Clinical Oncology paper, that he published in last year, and a couple of other directions we are working on incorporating these kind of models for doing drug response. And this actually, I realized much later that I just gave a talk on this, I think a few months ago at Texas A&M, and this is a work which we call, basically, let me go there. What we call an IRX model, an individualized therapeutic index model, that's been developed by my former postdoc Abhishek Minjin and current postdoc Satvik, who I believe is on the call, who's also a former Aggie, and a paper that's hopefully about to come out in AOS sometime this year or next year. I'll skip this part and now, okay, now get to the summary. Let's see. Yeah. So in general, just pulling it every together and thinking, what are the future aspects now? If I haven't conveyed integrative analysis, I mean, it's in general a very germane area in our work, and borrowing strength from these information-rich multimodal data structures and also biological knowledge. Like we haven't even scratched the surface in terms of the statistical and computational challenges, because as the volume of data increases, for example, in this case, we were just looking at bulk data with single cell data, which is an orders of magnitude higher. There's substantial analytic as well as computational methods that need to evolve. And of course, technology changes faster than the research can keep pace. Other types of omics, with CRISPR and microbiome, single cell, and also longitudinal. Now that the costs are coming down, essentially trying to hit the question that how does genomic diversity impact phenotypic diversity, and finally, of course, like sitting in a discovery domain, it's a very comfortable space. Discovering something, developing methods, but how do we actually get it to the clinic? And that sort of is the holy grail of how to actually do validation of that either in a laboratory setting or hopefully down the line in a clinical setting. That's at least a pipe dream at this point. So where does the future lie? At least in my opinion, it's integration and then innovation and of course implementation as best as we can do. So I want to acknowledge a lot of folks. I've shown you I've covered a lot of ground here, and this it took a village to do all this work, as you might imagine. So the background image, what you see here is my office in the School of Public Health, this yellow dot. So this is the School of Public Health. We have two buildings here. And this is where the biostat department sits, in this section. And my office is sort of on the bridge here. And this is where all the biomedical collaborators sit, this is where the cancer center is. And I wanted to show this picture because it's literally and metaphorically that this bridge had to be crossed multiple times, to get these things done. And I'm indebted to my collaborators, both at MD Anderson and at Michigan. And of course, on the top of the list is the folks I've worked with, both my trainees and students and postdocs I've worked with, as well as my biostatistical collaborators, and I want to say now mentors, especially Jeff, Ray, Bunny, and Keith and Kevin. I'll stop there.
Thank you so much, Veera. This is awesome. We have a couple minutes, maybe. I don't know if you want to entertain any questions.
Yeah, I'll be happy to. I'm trying to see if I can get the chat screen open. I ended up hiding it.
Right now, there's nothing in it, but.
Okay.
Does anybody have any questions they'd like to ask Veera?
See if I get, yeah, there, right. I see one.
Yeah, it's a just a thank you.
Yeah, so that's a good question. So are these priors from published literature papers? Some of them are. One of the particular priors that we use, the normal gamma prior, was developed by Jim Griffin and Phil Brown. But we extended it to another setting where we have group level selection, which we call a hierarchical normal gamma. That is something that Elizabeth developed during her dissertation. And so short answer to your question, it includes both. It's building up on these class of shrinkage priors for specific models. And is there any choice behind set these priors? I mean, the name of the game is getting sparsity and shrinkage. And that's why we settle on these choices. When you get into the high dimensional setting, which is the n less than p setting, You need these class of priors to do some amount of shrinkage and also get sparsity, especially from an interpretational perspective. And I know there's been a lot of both theoretical and empirical research showing the utility of these priors, especially for high dimensional regression. So I don't know if that answered your question, Jonghyun, but I can be more specific if you need me to be.
I could answer his question.
Hi, Veera.
Hey, Minsuk.
Hey, I have a question about I'm working on shrinkage prior and Bayesian model selection, and so when I implement Bayesian model selection or variable selection, the biggest challenge is how can you choose the shrinkage level, like tuning parameter selection. We cannot use cross validation. Then some people impose some hyper prior on that, but it does not solve the problem at all because hyper prior would have a hyper parameter again. So how do you choose the tuning parameter and hyper parameter?
Yeah. So let me go back to that slide. I can show it to you, specifically. We ended up actually having a hyper prior. And yeah, so this is the pathway version, but let me go back to the simpler version because the concept is the same, right? So if you think about it, just think about a regular regression, right? This is your betas, the betas form a normal, right? And this is the scale mixture representation, right? And then you have each of the scales has, so this is in a hierarchical setting because you have, platforms and genes, right? And then these would be the hyper priors, right? So these need to be fixed, right? So this is where I think it does take some amount of, for lack of a better word, sleuthing to get these parameters right. It depends on the degree of shrinkage. If you're going with shrinkage priors, that is, right? I mean, variable selection priors, it goes completely in a different way, but at least for the shrinkage priors. And to tell you honestly, as long as this is in a reasonable range, it actually works pretty fine, if you have seen. No.
I see. So it's a pretty robust to the choice of that hyperparameter.
Yeah, you can't go totally wonky with it, in the sense that you have to situate it somewhere where it produces reasonable values, at least of the scale parameters. Otherwise, it can break down. But as long as it's not too wonky, that you can. I'm forgetting the exact values, but there's actually a nice recipe that Jim and Phil Brown came up with to set those parameters, and that we used that as a default, and it worked pretty well. Basically, it's some kind of empirical based type situation where you can tune these hyperpriors. And it's, I want to say, I don't want to make a broad statement that it's not sensitive, but it's relatively, not that sensitive to this setting.
Okay, thank you.
And if you see it, these parameters are actually what, 1, 2, 3, 4 levels away from the data. So
is it mixing well? Mixing well?
I mean, in our simulation examples, yeah, it works pretty well. So this is essentially, you can think about this as a spiced up lasso, right? A lasso would be an exponential right here, right? The gamma, if you replace it with an exponential, it's just a lasso. But the gamma has 2 parameters now instead of being an exponential. And
it becomes a fatter tail. Hello?
Yeah.
Yeah, okay, thank you.
Did that answer your question? Yeah, we haven't tried the neuronal priors. Someday.
That results in the exactly same one. It just improves the computational efficiency.
Okay, well, Vera, I think that we're a few minutes after 1. Maybe we should wrap up, but it seemed like in your talk you were willing to share your slides and then also maybe even discuss questions with others later. So thank you so much. It was an excellent.
Thanks, Marcy. I see one more question came in. Do you want me to just real quick hit from Lisa?
Sure, I missed it. I didn't see.
A general question: Have have any of your research work helped in developing cancer drugs yet, or helped in precision medicine? So I hope I could say yes, Lisa, not yet, not yet. And I'll underline yet. And this is sort of a work in progress, as I said, more from a discovery standpoint. And what we are hoping is we can engage with, more collaborators and clinicians, to apply them. So right now we're still living in a discovery space. No, that would be the short answer, but we hope to someday.
Okay, well, thank you so much.
All right.
Have a
Thank you.
good day.